Okay, let's start together. Previous lecture will be recorded, but there is some mic issue, so audio is not available in this video, so I will record it second time. Okay, let's start. GPCR signaling. What is GPCR signaling and why it is important? CSIR asked question in GPCR signaling and RTK signaling. So for the CSIR purpose, it is very important topic. Now, why it is required? In this lecture we will learn why it is required and when it is required. If GPCR signaling is required, then what is the requirement of the body or cell, and when it is required. Okay, let's start without wasting any time. The topic is GPCR signaling. Topic GPCR signaling. Okay, what is GPCR signaling? First we will learn what is signaling. Okay, animal body is made up of different type of cells and collaboration of different type of cells. Cells in any animal body communicate with each other and make functional our body. So here is some cell and here is different type of, these are different type of cell. This is cell number 1 and this is cell number 2. So here are some cell producing hormones and chemicals, and here are some cell which produce which accept this chemical and hormones or signaling molecule. Okay, there are different type of signaling. The first, paracrine, autocrine and juxtacrine. This is not the topic. The topic is GPCR signaling, so we will focus on the GPCR signaling. The cell produce the signal is called signaling molecule. Signaling molecule. This cell is receiving this hormone, receiving this signaling molecule and functional activity perform. Okay, let's start. What is GPCR signaling? GPCR means G means globular, PCR globular coupled receptor signaling. Okay, GPCR phosphorylation. Okay, in GPCR signaling there is 3 main components. The first is receptor, which is seven-transmembrane receptor. That means it will cross the plasma membrane 7 times, like 1, 2, 3, 4, 5, 6 and 7. This is the C-terminal and this is the N-terminal. The first is receptor and the second is trimeric G protein. Trimeric G protein. The third is effector enzyme. Third is effector enzyme. These are the main component of receptor enzyme, main component of GPCR signaling. The receptor enzyme, trimeric G protein and effector enzyme. Then we will learn the epinephrine signaling. Epinephrine or adenylyl signaling / adrenaline signaling. epinephrine or adenylyl signaling. Okay, let's start. In epinephrine signaling, here is plasma membrane. Here is receptor, which is 17, 1, 2, 3, 4, 5 and 6. 1, 2, 3, 4, 5, 6 and then 7. This is the receptor. Hormone come here and bind here. Here is some protein, that is trimeric G protein, in which 3 subunits alpha, beta and gamma subunit are present. Then here is an enzyme molecule present, which is called effector enzyme. In epinephrine signaling, it is called adenylyl cyclase. That is adenylyl cyclase enzyme. Adenylyl cyclase enzyme. Okay. Whenever this epinephrine molecule is not present here, here is N-terminal. This is the N-terminal. This is the C-terminal. This alpha subunit is bind with the GTP form, GDP. When hormone is absent, this is the structure, that is receptor, then alpha subunit, beta subunit and gamma subunit. Now, what happens when receptor is bind? Here, receptor is bind, that is epinephrine hormone bind here, and then this is the subunit. Now this beta and gamma subunit is present, but this alpha subunit migrate here to here, that is adenylyl cyclase enzyme. This alpha subunit migrate here in form of GTP. Alpha GTP. Let's focus on this. Here it is present in GDP form. Now it will change into the GTP form. It is the active form of alpha subunit. Alpha subunit की active form है. Now adenylyl cyclase changed ATP to AMP to cyclic AMP. Okay. This cyclic AMP bind with the pKa subunit. Now, what is the function of pKa? There is two functions. It will goes into the nucleus, inside the nucleus. This pKa activate the CREB, 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 and this CREB will enter into the nucleus. This is the nucleus. Enter into the nucleus and bind with the Cre subunit of gene. Here is some subunit present called Cre. CREB come here and bind in this position and regulate the other function. The second pathway, it will contain 2 regulatory subunit and 2 catalytic subunit. This cyclic AMP, 4 cyclic AMP bind here, 1, 2, 3 and 4. There are 4 cyclic AMP. This is cyclic AMP bind with the 2 catalytic subunit and 2 regulatory subunit. Now, what is the function of PK? First, it will stop making glycogen molecule. Stop making glycogen molecule. Regulate the phosphorylation kinase, which will inactivate glycogen synthase. That will help to produce glycogen 2 glucose 1 phosphate, and this glucose 1 phosphate change into the glucose molecule, and this glucose molecule is basically the let me have new slide. Okay, this glucose molecule will enter into the cytoplasm of cell, then enter into the mitochondrial matrix, and perform Krebs cycle, and also electron transport system in this and produce ATP. Now, why this ATP is important? Why all this pathway is running? Why it is required? This is the question. Okay. So, sometime animal need energy instantly. Sometimes animals need energy instantly. So, in emergency situation, brain produces signal for making adrenal hormone. Some cells produce this adrenal hormone. These hormones are produced by few cells. Now, this is going to the bloodstream and then accepted by the receptor cell. Now, this is bind with the receptor cell, and inside the story of this receptor cell is here. When hormone is not bound, there are some 7 transmembrane receptor, that is alpha, beta and gamma is present here, and alpha subunit is form of GDP form. Here is enzyme adenylyl cyclase enzyme. Then if hormone is bind here, it will cause the alpha subunit change into GDP form to GTP form. This is the activated form, and alpha cyclase change into the cyclic AMP, and then PK produce. This PK is enter into the nucleus, then activate the crave, and then go to the Cre side and bind. And here is some other pathway, that is stop making glycogen molecule, because if we store glucose molecule, there is no instant energy for production, and we will produce glucose molecule by using glucose 1 phosphate to change into the glucose molecule. Then glucose molecule enter into the cytoplasm, then mitochondria, and then with the process of ETS it will produce the ATP, and this ATP is the actual form of energy or source of energy in the animal cell. This is the diagram. This is the signaling molecule. This is the receptor. Here is gamma and beta subunit is present. Here is alpha subunit present. These alpha subunits go to the effector enzyme in epinephrine signaling. In epinephrine signaling, effector enzyme name is adenylyl cyclase. Adenylyl cyclase enzyme, and this change into ATP to cyclic AMP, then pKa, then CRAVE, then it will go to gene transcription and regulation. This is the pathway, and here are some other pathway. We will not discuss about it right now. This is the GPCR signaling. Actual concept of GPCR signaling. The question will ask, what is the activated form of alpha subunit? What is the name of adenylyl cyclase? What is pKa done? How many units are in here? That is 4 subunit. These are the questions which will ask in the CSIR NET examination. Thank you.